Hi, everyone, and welcome to episode 781 of People Behind the Science. This week, we are rebroadcasting our interview with our guest, Dr. Patrick Schloss. Listeners, Pat is a microbial ecologist who studies bacterial communities. His work examines how the bacterial communities in us and on us interact with each other and with us in order to keep us healthy. And then conversely, also how to get these bacterial communities back on track if something goes wrong. Pat has also been building cool bioinformatics tools for computational analyses. And in our interview, we talk more about his exciting research and share stories from his life in science. So listeners, get ready to sit back, relax, and enjoy this episode of People Behind the Science.
Every day, discoveries are made that will change our understanding of the world around us. Dr. Marie McNeely is here to bring you the brilliant minds who are making these discoveries so they can share their incredible stories and take you on an amazing journey. Welcome to People Behind the Science.
Hello, everyone, and welcome to another episode of People Behind the Science. I am thrilled to be here today with Dr. Pat Schloss. So, Pat, welcome to our show. How are you?
Great. Happy to be with you today.
Well, we are happy to have you on the line with us, and I'm going to introduce you now to our listeners. So, listeners, just a little bit of detail about Pat. He is an associate professor in the Department of Microbiology and Immunology at the University of Michigan. Pat received his bachelor's and PhD in biological and environmental engineering from Cornell University, and he then went on to conduct postdoctoral research at the University of Wisconsin and served briefly on the faculty at the University of Massachusetts Amherst before joining the faculty where he is today at the University of Michigan. And now, listeners, Pat is here with us today to talk about his experiences in life and science. So, Pat, we want to get to know you today, both as a scientist, but also as a person. So can you start off by giving us an idea of how you spend your time when you're not doing science?
Well, I guess the thing that sticks out most about me is that I actually have 8 children between the ages of 14 and 2 months old.
Oh, wow.
So there's not much time for me to do things that I necessarily want to do. So kind of my interests and my kids' interests overlap a lot. And we own a farm that's about 20 minutes outside of Ann Arbor where we've got sheep and cows and pigs and chickens. So when I'm not at work, that's what I'm doing is taking care of them. And we just got off a week at community fair nearby where I worked with my kids to show their animals. And that's an activity that we've really loved doing as a family, because I see a lot of my colleagues who have kids and they're taking each of them to a different activity and just spend a lot of time driving around in a car.
Right. You're the chauffeur. Yeah.
Yeah. I mean, that's just like the worst. So this is something we all get to do as a family, and it's hard work, of course, but it's a lot of fun, and it's very satisfying to sit down at the end of the day. And in the summer when we've got a garden going, to have the kids point out that everything on our plate came from within a mile of our farm, from our neighbors or from us or from whatever. And the way I got into this beyond the kids was that going through graduate school and postdoc and being an assistant professor and moving to Michigan and just everything being really intense and constantly thinking about science, which is a good thing. But this was something that I picked up as a way to not have to stress out about science all the time. And so much of what we do in science, we get to the end of it, I can submit a paper and I can tell you every problem with the paper. I can tell you exactly what the reviewers are going to say. Mm-hmm. My own worst critic, right?
Right. You've got the responses ready.
Yeah, right. But I mean, if I sit down and I'm having a steak that came from my son's steer, and that's perfection. There's really no revisions that you need to be done. That's something that is kind of an objectively good thing. Whereas so much of what we do in science is, I don't know, not to be negative, but negative.
Right.
We're never going to get the absolute truth of what's going on. We're always kind of feeling around in the shadows to some extent. And so for me, it was really nice to have this outlet where I don't have to be a scientist. But of course, we'll talk about what I do. I study bacteria and poop, and I can't help but walk through a pasture and kick manure around and think about the bacteria that are in the manure. So
you're never truly separated from the science.
Exactly.
Well, Pat, wonderful to get a window into your life outside of science. I think it is great too, like you say, have this place where you can go to escape a little bit, to get away from the negativity sometimes that you're immersed in the world of science, all the people nitpicking holes in everything that you're doing. So we are going to talk about the science next. You alluded to it a little bit. You study bacteria and poop. Tell us a little bit more.
So as you mentioned, my training in engineering got started thinking about waste treatment of all things and thinking about bacteria that are active in compost and wastewater and kind of gradually have taken those ideas of understanding how bacteria work as a community to break down waste. And what I like to think about is the idea that if you're familiar with the book Horton Hears a Who from Dr. Seuss, he picks up a flower and he realizes there's a little community there, the Whoville. Mm-hmm. And so the way I think about it is that within us and on us are all these Whovilles of bacterial communities interacting with each other and with us to keep us healthy. And if something goes out of whack, then we need to figure out what that was about and what's going on, and can we perhaps remediate them to get back to where they can confer health. And so that's really what we're about, is trying to understand how these communities of bacteria that live in and on us keep us healthy.
Very interesting. Well, I was curious about that engineering connection, so very cool.
Yeah, and a lot of what we do does have a pretty strong computational angle to it. And when I'm not doing the microbial ecology work, as we call it, we also do a lot of bioinformatics work. And so when I was a postdoc, we had all these great questions that we wanted to answer, but we didn't really have the computational tools to answer them. Just really simple things that we needed. And we kind of looked around the lab, and I was the one that knew how to write a computer program from my training. And that skill has really served me well, because now a lot of what we also do is this bioinformatics work and tool development that makes it so people can use these tools. So it all makes sense. But when you get the long explanation, when you just look at the CV, right, sometimes it's not so clear.
Raise an eyebrow a little bit. No.
Yeah.
Well, wonderful to hear a little bit more about your science. And now we've heard a little bit about your life outside of science as well. And I must say, I'm very impressed at how you're able to get it all done. 8 kids, managing a farm, doing research, being a faculty member. It is a lot to get done in a day. Do you have a motivational quote or a saying or a force that really keeps you going and allows you to do it all?
It sounds good, but sometimes it's pretty chaotic. But I'm like a lot of people and get very distracted at work. It's very easy to just kind of start going down through emails or browsing links from paper to paper or website to website, or getting sucked down a hole in social media. But I force myself to frequently think about, do I really need to be doing this right now? And if I look back and realize I just wasted an hour, I can kind of guilt trip myself and think, that was an hour I could have been at home outside with the kids or doing something.
Ooh, that's good.
Yeah, a pretty big guilt trip that I can lay on myself. But at the same time, it just allows me to then say, hey, no, I mean, I have stuff I need to do here, so let's get that done. But that's one of my big motivations at work for trying to be more productive. But admittedly, there's so many distractions, many of them good, but sometimes we just need to get to work.
Just pure distractions, yeah.
Yeah.
Gotcha. So is there a particular quote or saying that resonates with you?
For science, there's this quote that I think is from Jacques Monod. He said something like, what's true for an elephant is true for E. coli, only more so. And I think he interpreted it in terms of perhaps enzymology or biochemistry. What I interpret it is that you think about the ecology of an elephant and how it interacts with its environment out on the savanna or wherever it lives. And E. coli and all the other bacteria out there are very similar in their ecology, that they have an ecology where they're interacting with other organisms. And so a lot of what we like to think we do is to take ideas from ecologists that study plants and animals and try to see if we can apply those to microbes. And so thinking about things like competition and predation and parasitism and symbiosis and evolution, and trying to couch a lot of what we do in the lab in terms of ecology. We work with clinicians a lot. So when we start talking about antibiotic treatment is kind of like a forest fire.
Clears everything out, yeah.
It clears everything out. And then we start talking about Yellowstone recovering from forest fires, or the response of Yellowstone to reintroduction of wolves, or things like that being similar to what we're trying to do with probiotics or Clostridium difficile infecting people. They kind of like are taken aback because that's just to them a very different way of thinking about things, but that's really how we think about things. And so that idea that an elephant and an E. coli cell really aren't that far apart. That's one of the scientific quotes that I like to think a lot about, and kind of is a hypothesis that maybe we can test through our work.
Well, this is a fantastic quote and a really interesting perspective on the world, applying some of these same ecological principles and concepts now to the smaller things that we can't see, this whole hidden world to us. So very, very cool to hear about some of these things that keep you motivated, keep you going in your hectic daily life. But now I want to talk a little bit about some of the people who've helped you along the way and kept you going through your scientific career. So can you share with us a little bit about some of these role models or mentors that you've had along the way?
I think any scientist that says they don't have a role model is lying. And I've been very blessed to have people throughout my life that have encouraged me and made science fun and inspired me to keep going further. And somebody that really stands out to me was my postdoc advisor, Jo Handelsman. She's previously at the University of Wisconsin, and then she went to Yale and is now working in the White House in the Office of Science and Technology Policy. It's funny because I, this past week, had the opportunity to visit her in Washington, D.C. and have lunch with her in the White House.
Oh, wow. When
I walked into her office, it was like a flashback, like I was walking back into her office as a postdoc to talk about data. It was pretty wild, but she's just been, to me, such a great cheerleader for my science and the science of others, and really does a great job of thinking about ecology and thinking differently. And there was a time when I was working on something in the lab and it just was not working. And she just popped her head in my office and said, "So, hey, I haven't talked to you in a couple of weeks. What have you been up to?" And I had this kind of crazy idea that I was working on to try to get around it. The problem was trying to figure out how many types of bacteria are in a gram of soil. I just couldn't wrap my head around something. And so I said, I just can't figure out this estimation thing. I've been trying to estimate the number of words used in a book as an analogy for that. And I've been using this book called Goodnight Moon, which, if you're a parent, you've probably read it 1,000 times to your kids.
It is an excellent book.
Yeah. It has like 151 words and I think 32 total different words or something.
Sure.
And so I just kind of told her this silly idea that I'd been working on. And she's like, "That's brilliant. My favorite book is Portrait of a Lady. Could you analyze that for me and show me what the sampling curves look like to figure out how many words were used in that?" And so what I thought was just something really stupid, she thought was brilliant and turned into a paper. And I now know that that paper has been used by many people in teaching undergraduate classes and getting them to think about issues of sampling. I mean, she's just been really good throughout my career, but especially as a postdoc at being very supportive. And also, when I went to her lab as a postdoc, I thought I was going to go there to do science. And she was the person that coined the term metagenomics, which has really driven a lot of modern microbial ecology. And it turned out that a big part of what she also does is educational and thinking about how we teach people science. And the idea that she brought to me and to many others was the idea that we're scientists, so we should teach like scientists. And that doesn't mean that we get up there with a pocket protector and lab coat and talk really boring.
Right. Nerd it up. Yeah.
But let's form a hypothesis about how people learn. And let's form a hypothesis about the best way to communicate some idea about microbiology. And let's go into the classroom now and test that by how we teach. And that just really blew me away. And it affected how I went on to do my job talks, because I did a job talk where, similar to sampling words in a book, I had people sample M&Ms out of a bag. And so a formal seminar, I'd go around with bags of M&Ms and brown paper bags and have people think about, well, how do you define a type of M&M, or how do you define a type of bacterium, and how many candies do you need to sample before you know all the types that are there? If you've ever been to an academic seminar, that is not the way they're done.
Right. But they should be. I like this chocolate. And yeah.
Exactly. But 10 years later, people are like, "You gave a talk where you handed out M&Ms," and that stuck with me. I never would have done that if Jo hadn't been so encouraging and said, "Yeah, I mean, it's weird, but people are going to remember you. And part of research is that we've got to teach people what we're doing." So when I think back about people that have affected me and influenced my career, she's number one.
Well, that is phenomenal. Excellent to hear you had this great mentor along the way to teach you science, also teach you the importance of this educational component. And I think it is so important. So has her passion for teaching rubbed off on you?
It has, although I'm in a med school where we do very little classroom teaching, but I still look for ways to do more informal teaching around a lot of the bioinformatics we do. And that is a subject where people are very scared to interact with a computer to analyze millions of DNA sequences. And so I'm always looking at ways to improve how I teach and thinking about being a scientist, how I teach this, what's a hypothesis I can form to teach that better. And let's look at the literature and think about how we can change that. So it definitely does stick with me. And as my kids are in school, I also can think about how their teachers are good or could do better. So it's
You're doing the home education as well.
Yeah.
Well, Pat, wonderful to hear about one of these people who had a really big impact on you and your career. But I want to go even farther back now and hear about what started you on this path to science to begin with.
I grew up in Columbia, Missouri, which is the home of the University of Missouri. And while I was there in junior high and high school, I had just phenomenal teachers. And what really stuck out to me were my math and science teachers, and that We did science fairs. We did very simple experiments that my parents helped me set up and run. And the teachers were just great in encouraging us, getting us excited about science. And a couple of months ago, there was a thing going on Twitter with a hashtag called I am a scientist because. And I don't know, I was feeling sappy at the moment and thought, I'm a scientist today because of Mrs. Pickett and Mr. Burns, who are my biology and calculus teacher. And I thought, sure, I could send out a tweet saying that, but that doesn't mean much to them. So I actually sent them an email telling them what I'm doing and how specific things in their classes still stick with me. And I think they were a bit blown away by that. And thankfully, I think they remembered me, but
if not, they pretended well. Yeah.
They pretended very well. And so I think it's important that people think about that question, but then also act on that. Certainly those teachers in biology and math really stuck with me. And when I went off to Cornell for undergrad and grad school, I had difficulties getting into graduate programs. And so I think this is also a common story where many of us faculty did not get into our grad program of choice and didn't necessarily do very well in high school or undergraduate career. And I went to my undergraduate advisor, who I'd done research work, Larry Walker, asking him for another letter of recommendation to send out to one more school just to see if I could get in. And I think I just paused and said, Dr. Walker, tell me about what you're doing. And he could have told me that he was studying foot fungus, and I would have thought it was just the most amazing thing ever.
Right.
I think at the end, I just said, don't worry about that letter. Is there any way that I could do my PhD with you? He was studying activity of cellulases and breaking down cellulose and composting, which are not the sexiest scientific subjects, but I've done very well by having studied those things. And so he was there for me. I applied to like a dozen grad schools and got rejected from all of them except for Cornell.
Yeah, you had the foot in the door.
I had the foot in the door, right? And that worked out really well. And so things have trickled from there, going to, like I said, Joe Handelsman's lab and moving forward. There's always people that inspire us and look out for us, and that's really been really helpful.
Well, thank you so much for sharing this story. I think it's really important for people out there to realize that even if they maybe didn't have the grades that they hoped for in high school and college, you can still go on to be a successful scientist. I think this is so important. You have to be persistent. Maybe you have to make connections and be a little more strategic about it, but you shouldn't let that stop you.
Absolutely. And it's surprising now that I'm on the other side of the table that at one point I wanted to be a doctor. I wanted to go to med school. And I took the MCAT, and it was a horrible experience, and I didn't have the grades.
I'm sure people are commiserating out there with you.
I don't know what I was thinking in trying to go that route. And now I'm a professor in a med school, and I can see what the first year med students are like. And it's like,
right.
They're just as normal as or weird as I was when I was a first year grad student. And then being on an admissions committee, the thing that we all say is that, man, how did any of us get into grad school? And so that's something I worry about a little bit, that things are so competitive that I wonder if we're losing the Pat Schlosses of the world that had a 3.2 GPA in undergrad or didn't have the best research experiences necessarily. Because students that come in with a 4.0 aren't always the ones that are the hardest workers or can make the weird connections that help propel our research forward. So it's not a straight line.
Definitely. And I think even more so now that I'm on the other side as a faculty member interviewing faculty candidates, you're like, how did I ever get this job?
Exactly.
People are becoming more and more impressive, I think, as it gets more competitive. But I think so important to get that message across that the doors are open for a science career, regardless of the path that people take to get there. And now I'm curious, we left off here. So you've gotten into graduate school at Cornell. Can you tell us a little bit next about some of the key moments that then took you from that point to where you are today?
Yeah. So in graduate school, I studied composting and kind of what the bacteria were doing to drive that process. And one of the things that we did was we had this very artificial system to control as many of the variables as possible. And so you can imagine, if you're composting food waste, there's all sorts of variation in what the wastes are. And so what we did, we used dog food instead as our model food waste. And that's just one example of how contrived we made the system. But even though we made this really contrived system, we still had a lot of variation between implementations of the process. So if I set up a compost reactor and ran it today and then ran it in 3 weeks, the performance might be very different from run to run. To an engineer, that's just a horrible thing because engineers, we need to control things.
Right. It needs to be precise.
And so my hypothesis was that it was about the bacteria, and that because somehow we were getting different types of bacteria into the reactors, we were getting different process performance. And that turned out to be right, that as you change the seasons, there's different bacteria in the air that are coming in to seed the reactors, and that causes variation. And so that then led me to think about, well, we know who's there, what type of bacteria are there, but we don't really know much about what they're doing. So my push to postdoc and working with Jo Handelsman was that she had developed a technique called metagenomics where they could capture all the genes from the bacteria in a community and then characterize those genes. And the challenge with studying bacterial genomics is that we can only culture about 1% or fewer of the bacteria in a community. There are entire phyla of bacteria that are not cultured. So the analogy I give to people is like, imagine you're a Martian looking down on Earth and you say, there's this green stuff down there on that planet, and we don't really know what it is, but all that green stuff is kind of similar to each other, and we don't really know what it does. And we think of those as plants. Mm-hmm. Just imagine there's this thing called plants that we don't know anything about, but we see evidence of it everywhere. And that's par for the course with bacteria. There's entire phyla of bacteria that all we know about is based on a couple hundred bases of their genome, and we know nothing about what they do. And so she was developing techniques called metagenomics to get over that without having to do the culturing. So I switched from compost to her lab to studying soil and working on some projects in her lab looking at insect guts and gypsy moths and thinking about ecological ideas like diversity and stability in communities and developing bioinformatics tools. And from that, my plan was, I think, to have a lab fairly similar to hers as a professor. And that's when I went off to the University of Massachusetts in Amherst as an assistant professor and really ramped up our work in bioinformatics and developing software. And so we developed a software package called Mother, MOTHUR, and using that to facilitate research. So that was published 5 or 6 years ago, and it's gone on to be cited over 2,500 times, which is amazing. Yeah. It's a very simple concept of what it does.
But so useful.
But so useful, right? That's kind of where I started to make a name for myself, I feel, is with those bioinformatics tools. And there I was studying some things with soil and some things with insects, but nothing really got going at UMass along those lines. And at about that time when we'd put Mother out and were kind of getting the biological stories going in the lab, the NIH released a broad project called the Human Microbiome Project, which had the goal of characterizing the bacteria that lived on and in the human body. And I went to a study section, which is kind of how NIH reviews grant proposals for this project. And I noticed that my name wasn't on any of the proposals, that I wasn't asked to be a collaborator on those. And that kind of annoyed me because I thought, I do pretty good work with bioinformatics. And if I think about where I'm at, there's really no one around me at UMass doing that type of work. There's no one doing big DNA sequencing. I don't know that we even had a sequencer at UMass. And so I was developing bioinformatics tools, but didn't really have colleagues there to apply those. And as I was sitting on the airplane going back home, I opened up an issue of Science and saw that Michigan had an ad for 3 positions, all within the area of microbial ecology. So they wanted somebody in epidemiology, they wanted someone in microbiology, I guess there are 4, someone in ecology and someone in molecular biology. And I said, man, we just reviewed maybe 20 or 30 proposals. And if any of those proposals had all 4 of these people, it would have just been out of the park. It would have been an awesome proposal.
Sure.
And so I applied. And it was funny because the person that I talked to about it, Vince Young, who is now at Michigan, he's like, oh, you saw that, did you? We kind of wrote it with you as our muse, so to speak.
Really?
And so I applied, and it was very competitive and somehow got the position. And it's just been wonderful coming to Michigan. And I really enjoyed my time at UMass, but the University of Michigan has really invested a lot of resources into the Human Microbiome Project. And out of those 4 initial positions, through things like spousal hires, also just identifying good talent, I think they ended up hiring about 8 or 9 people along that came out of those initial 4 positions. There was already a lot of strength here in that area of research. So it's been great to be at Michigan because of great colleagues and great resources and great opportunities. So that kind of brings me to where I am today, where we continue to do a lot of the microbiome research, looking at the role of the microbiota in colon cancer and in allowing this pathogen Clostridium difficile to colonize. And I have opportunities now that I never could have imagined. I've got co-authors that are physicians, and we're doing a lot of what you might call translational research, where we're working with human subject samples, we're running animal models using mice and things like that. So it's been a long way from composting
Sure.
to the human gut, but along the way, thinking about things like dynamics and reproducibility still fascinate me. Just like I could take 5 compost runs and say they don't reproduce each other very well, I could take 5 people, sample their gut microbiota, and they're totally different. And we have no idea why. So it's very different system, but very similar questions. And that's one of the things about microbial ecology and microbiology in general that's so fascinating to me.
I definitely agree. This is a very complex and really interesting system that I think is getting a lot more attention now than it ever has in the past. But I always have this sort of nagging question in my mind. Do you think that there is an optimal microbiome, or we'll say gut microbiome?
No, I don't.
Really?
Yeah. So this is kind of a controversy or an idea that's around in kind of the popular thinking about the microbiome.
I'm always looking for something to aspire to. Yeah.
Yeah. People talk about a healthy microbiome, and we could line up, and people have done this. So you line up 100 people and you sample their gut microbiota, and there will not be a single species in those people that is shared across all the individuals.
Wow.
And so there's this huge variation. And yet all those people are healthy, quote unquote, whatever healthy means, right?
Sure.
So I think our challenge is to figure out what makes somebody healthy. And also the idea that what might be healthy in one situation might not be healthy in another situation. So you could have a community that makes you susceptible to Clostridium difficile infection, but might protect you against colon cancer. And you can play that on with diabetes and obesity and all the way out. But that there's a lot of variation in what these organisms are doing and how they come together. And we really are looking for some unifying principles about how can you have all this variation yet still have health, and thinking about what situations allow different people to be healthy or not. And what we really think a lot about is, again, the variation. And my vision of the future would be that every year I take a stool sample to my physician, and she calls me and she says, "Hey, Pat, we noticed that your community has really changed since your last visit. We want to get you in for a colonoscopy, or we might want to check your blood glucose level, or do these different tests based on the change in my community." Not necessarily comparing me to you, but comparing me to me a year ago.
Over time, yeah.
Because we know that, again, if I took those same 100 people that we sampled before and sampled them again in a year, each of them is going to be more similar to themselves than they'll be to any of those other people. So I think about health perhaps much more on kind of the individual level than on a human population level.
Certainly. And I think there's always the possibility that, similar to ecology, there are a variety of different species filling the same role. So if you have one of those, you might be okay in certain situations.
Absolutely.
Well, great to hear about your work, Pat. But I think we've painted your journey in science as a little bit of an adventure. We talked about some of the stumbling blocks along the way, but I think it is so important to bring these to light, because I think a lot of people see just the success stories in the media. They don't really appreciate all the challenges that scientists face in their daily life, but also throughout their career path. So can you tell us today about one of your own, maybe a major failure or a challenge or a frustrating situation that you faced and how you got through it?
I once had a student that was annoyed that lab experiments weren't working, and she thought it was some reflection on me as a PI. And I was just like, no, I mean, that's just science.
Right. You could blame me, but it's science's fault.
But it's science's fault, right? So I feel like I had a hard time landing my first tenure track job at UMass. And I think that's kind of a common concern of a lot of grad students and postdocs. I was on the job market, I think, every year that I was a postdoc. And in engineering, people didn't do postdocs when I came out of grad school in about 2002. So my last year of grad school was on the market, and then I maybe took a year off to be a postdoc. And then for the last 3 years at Wisconsin, I was on the market. So I probably might have been close to 80 or so applications that I put out there.
Oh, wow.
A bunch of interviews. And I took the first job I was offered. So I wasn't going to leave anything to chance. And along the way, I realized that I was making a really big transition from engineering to biology. And that was something that I didn't appreciate being as hard as it was. And it really is a different way of thinking about things. engineers, we solve problems, right? Like if you're in waste treatment, we've got this weird waste, we need to get rid of it. So there's a lot of case study driven things, whereas in biology, it's much more of big picture. I remember I interviewed for my dream job, which would be at Michigan State University, just up the road from where I'm at now. And this is the university that I consider to be the best place to be for microbial ecology. And just the who's who of microbial ecology were there. And I gave my talk, and I think it was okay. And then I did a chalk talk where you stand in front of a chalkboard, basically, or a dry erase board nowadays.
These are terrifying, by the way. Yeah.
Yeah. And nobody does them well. Nobody knows what they're supposed to do. And you kind of lay out your research vision. And Rich Lenski, who's just an amazing evolutionary biologist with E. coli, at the end is like, yeah, this is all cool. But like, what's your hypothesis? And what I'd been explaining was how I had used tools that we developed for doing statistical hypothesis testing. So we had this problem where we didn't really have a way to do statistics with microbial communities. So I built a tool to do it. Very engineering, right? Like, we've got a problem. I solved it.
Right. Textbook engineer. Yeah.
And I was kind of like, well, what do you mean, Rich? Now we can test a hypothesis.
I've solved it.
What do you mean it was my hypothesis? I just showed you I just solved it. And we kind of went back and forth a bunch, and I just had no idea what he was talking about or getting at. I thought he was just this crank. He's a very nice guy. And a couple weeks later, Tom Schmidt, who was the search committee chair, called me and said, hey, Pat, I just wanted to follow up with you. I'm sorry you didn't get the job. You were one of the finalists, clearly. But if you have a moment, I just want to talk to you a bit about your chalk talk. And he just broke it down for me, what Rich was getting at, and the idea that to a biologist, a hypothesis is kind of your big vision of the way the world works.
Right.
And that you're going to spend your career testing that hypothesis. And Rich just really wanted to know, like, what are you about? What drives you? What motivates you? And I just we talk about face palming now. Right. It was like a duh moment. And I am so grateful to those two gentlemen. I still struggle with this because it's so much easier to solve problems than to test or come up with some big grand vision. And now Tom Schmidt has actually moved down the road to the University of Michigan, and his office is 2 doors next to mine. And we still have great conversations. But I mean, I thought that was so magnanimous of him to help me out, right? And to mentor me. And I think once I made that shift in thinking, it still took me a little bit longer to get a job, but I could at least begin to think more in terms of biology and what was my hypothesis. So that was a big challenge that I guess I didn't realize I had until Tom and Rich called me out on it, but I was very grateful. So,
well, I love this story, Pat, and I think that is an important lesson. I think anytime you're transitioning between scientific fields, it is a challenge. There's a whole new language to learn along the way, but I think just the way of thinking between traditional science and engineering are often very different. And so this is a great example of that. So we've talked a little bit about one of these challenges, but I don't want to dwell on our negative times, our down days. So Pat, I want you next to share a success story with us, and it can be a big victory or just a small win that meant a lot.
One of the things that I've just been really impressed by is that I've been blessed to have some really just awesome students, both at UMass Amherst and here at Michigan. And I feel like the last 2 or 3 years, we've been on a roll just putting out some really great papers that we're really proud of. And again, we've been looking at things like colon cancer and Clostridium difficile infections. And one of the recent series of successes has been using a mathematical statistical approach called random forest modeling, where we've been able to take microbial communities and predict an outcome. And so one of the big criticisms against microbiome research is that it's all correlation or association, that there's no directionality, or that it's all soft science, so to speak. And so what we've been able to do in these different experimental systems is we're able to use this modeling approach to predict whether or not somebody has an adenoma or carcinoma in their colon based on a stool sample. And in mice, if you give me a community, I can tell you how many C. difficile will be in a mouse the next day after we challenge them with C. difficile, again, based on the microbial community structure. Or using a cancer model in mice, again, I can tell you how many tumors that mouse is going to have 75 days later based on the type of bacteria that's in their community today. And so these are, I think, pretty big advances in thinking beyond just kind of correlation or association, but moving more towards prediction and using these microbial communities to make more quantitative predictions about what's going on in the future. And these stories have kind of played out in a series of papers from my lab over the last year or so, and it's been a lot of fun. And I think sometimes there are PIs that there's always the one project they really don't want to talk about because it's just not that exciting or not going very well. But I've just been really impressed by my students pushing these projects through. And I think I annoy them because I'm constantly nagging them to like keep going.
Right. No time for sleep.
I think their impression is that I don't think they're working hard. And in contrary, I'm just really excited about this stuff, and I really want to get it out there, and I want people to see the great stuff they're doing. And so to brag a little bit, my last 2 PhD students are going to have 4 or 5 first authored papers from their work.
That's fantastic.
Which is amazing, right? My department has a minimum rule of 1 paper, and there are people that struggle to get 1 paper. So just things are really clicking right now, and that's really exciting. And I think also speaks to the interest people have in this field being a new area of research studying the microbiome. But certainly great students make it all possible.
Well, this is so cool. Now, I have to ask, in terms of this cause and effect relationship, do you think the different bacteria that you're seeing lead to the cancer, or does cancer change the environment and lead to different bacteria?
It's both, I think.
Okay.
So certainly there are bugs, bacteria, we call them bugs affectionately, but we put the bacteria in and they can then change the tumor profile. Or we can treat mice with antibiotics and we don't get any tumors. I'm not saying that people should go out and take antibiotics to prevent tumors, but we can change the community and change tumors. We certainly think that there's an interaction between the host immune system and the microbial community, so that the microbial community changes and that ratchets up inflammation, and then the inflammation changes the microbial community. And so it's kind of this bad feed-forward loop that drives inflammation and tumorigenesis.
Right.
And we've studied this more with mouse models, using antibiotics, using mice that have no bacteria and that we can then put in defined communities. So it's a bit of everything. And that systems way of looking at it, again, I think plays nicely with the idea of the ecology, in that it's no one thing. I sometimes drive people nuts because people say, well, what is the mechanism of colon cancer, the microbial role in colon cancer? I'm like, what do you mean, what is the mechanism? There's like a whole bunch of mechanisms.
Right. If only it were that simple. Yeah.
Yeah. And again, going back to that idea that everybody has a different microbial community, I mean, that's the same for everybody with colon cancer has a different microbial community. So each of them probably has a very different set of mechanisms. And so what we're trying to do with the mice and with studying human populations is kind of figure out, again, what are those commonalities and mechanisms or things that are driving and helping to move things along. But it certainly makes the research a lot of fun.
Absolutely. Well, congratulations, Pat, to you and your students for this phenomenal work. It sounds like so many exciting directions the laboratory is going.
Thanks.
Well, I love talking about successes with scientists, but I also love to get book recommendations. I am an avid reader myself, and I'm not sure if you can even fit time in your day to read. But I will trouble you for a recommendation here today, Pat.
It is hard to find time to read, but so I'm like a very avid podcast listener, which aren't books, but there's probably a dozen podcasts that I listen to a lot while I'm driving around or doing chores around the farm. I got an Audible account, which allows me to listen to a lot of audiobooks, which has been really nice. But the science book that I've read, or listened to it technically, that really drives a lot of my current thinking and reflection is The Immortal Life of Henrietta Lacks by Rebecca Skloot, which is the story, for people that don't know, of HeLa cells and the cell line that I don't use, but a lot of my colleagues use to study everything from viruses to cell biology and everything else. And what I've spent a lot of time thinking about is that when we study stool samples from people with adenomas or carcinomas in their gut, or people that have had C. difficile infections, that those actually come from people. That I think oftentimes as scientists, we get these samples and like, oh yeah, that's a stool sample.
Right.
But no, that's a person, right? Like, I mean, the stool isn't a person, but it came from somebody, and everything we get is de-identified now, probably because of Henrietta Lacks and the disaster that kind of transpired there. But probably a good number of the people that we got carcinoma samples from are passed away. There are real people that have these problems that we're trying to solve. And so it's just really gotten me to think more that we do science and we do research on these samples, but we're really dependent and should be very grateful and respectful to the people that we get these samples from. And so that's something that I try to think more about. How can we acknowledge these people and perhaps do a better job with how we communicate the science, because these are diseases that are not uncommon. And if we can educate the community better, then perhaps it's a way of repaying their service of providing us with these samples.
Certainly. I think this is so important. And I'm a scientist who works with patients myself. We work with people with Parkinson's, and even just being able to give them their own results after a study, or give them the paper that's published on the study that their data went into, I think is a huge sort of obligation that we have to keep people in the loop and keep people engaged in our research. So wonderful to hear about your book recommendation. I know we've had a couple people on the show mention it, which means I need to keep bumping it up on my list to read.
Oh, yeah.
Well, Pat, next I want to talk about science travel. You mentioned that you like to listen to podcasts when you're out and about in the car. Tell me about your favorite place that science has taken you.
I'm a weird scientist, I think. I really just hate traveling.
Really?
Yeah, I hate it. When I was a postdoc in Joe's lab, she had coined and initiated this really cool new technique of metagenomics. And so there was a lot of demand for people to give talks about metagenomics. And so I was that guy, and I was a 24-year-old postdoc that wore Birkenstock shorts and a T-shirt sitting up in first class. I was flying so much. And
yeah,
I mean, that was fun. But as my career progressed, it didn't really slow down. And I would travel once a month. I would try to limit myself to one trip per month. And I got to the point where with all these kids and my family and my farm that I sat on a plane and said, is anyone going to remember this talk 100 years from now or 10 years from now?
If there's M&Ms, maybe.
If there's M&Ms, they will, right? But I could maybe just ship them some M&Ms.
Right.
But my kids will remember that I'm home. So there's been really awesome conference invitations that I've had to decline because I need to be home. So I try to be really selective now about what meetings I go to. I went to a workshop in all places at Duke last year, working with a bunch of grad students and postdocs, thinking about reproducible research and what techniques or how do we teach people to engage in reproducible research. And I think the thing that made that trip so special for me wasn't that it was in North Carolina, which
it's a beautiful
no
place.
offense to people in North, is a beautiful place, but isn't like that amazing.
Sure.
Was the people that were there and getting to know these people and kind of continuing on those relationships and friendships. So I've been to like Estonia, I've been to Finland and some really objectively cool places, but I'm just not a big travel buff at this point. I've kind of been like, yeah, I did that, but
been there, done that.
Been there, done that. And it's kind of a little contrarian there.
Gotcha. Well, I think this is so important and an important point to get across to our audience. This balance between travel and family life and other obligations that you might have is something that you're going to face as a scientist. And sometimes it is hard to say no to those invitations.
My friends make fun of me for this. I went to a conference in Vienna, Austria, might be close to 8 years ago now. And I was walking around and there's all these gelato stands and I love ice cream. It's my vice. So I would go from like gelato stand to gelato stand. And I was thinking like, I'm all alone walking through this amazing city and could go to museums or do all these different things. But 10 years from now, I go to my wife like, hey, hon, remember that time I was in Vienna and walking, like,
eating gelato?
I'm eating gelato. Like it's not a shared experience. So my friends make fun of me because I went back to my hotel room and basically wrote a paper. And it's just like, you went to Vienna to write a paper, Pat? What is wrong with you?
And eat gelato.
And eat gelato, right? So that's just kind of the warped way of Pat Schloss, but yeah.
Oh, that is hilarious. Well, I really love that in these travel experiences, you keep pointing to the people, and I think the people in science make it fantastic. And I worked with so many phenomenal, hilarious, inspiring people over the course of my career. And I think this really brings a great personality to the different labs you're in. You get some quirky traditions that spring up and just share some hilarious memories together. So Pat, can you let us in on one of your own memories that demonstrates this human side of science?
Something that I try to do is go to lunch with my lab on a pretty regular basis. And I don't do it as often as I should or would like to, but we work in a hospital environment. And so we frequently go to the hospital cafeteria and eat lunch together as a lab. And my approach is, well, I've got to eat lunch, right?
Sure.
But when I start hearing students comment on that in their defense seminars, that they appreciated me doing that, it's like, I guess that's a thing. Now that I think about it, most PIs don't eat with their lab on a regular basis. And yesterday we ordered some pizza in and we did our fantasy football draft for the upcoming year.
Nice.
Which we have a picture of us really intently looking at our laptop screens. And I'm sure people walked by thinking, man, that's a group that's really collaborative and working hard together. And
sure. Such great science. Yeah.
They didn't see ESPN.com was on the computer for everybody there. So I think just having those types of interactions with people in my lab, and there is kind of a seniority scale in a lab, a boss.
Right. The hierarchy you can't avoid. Yeah.
There's a hierarchy, right? But I think anything I can do to get away from that as much as possible and get them to see me as their scientific colleague that we want to spend time together. And I mean, we're not going to spend all our time together, but the time we're going to spend together is going to be pleasant and have fun. And we'll talk about science, but we'll also eat pizza and draft our fantasy football teams together.
Smack talk, yeah.
And smack talk will go on for the rest of the fall.
Well, that is fantastic, Pat. It sounds like you have built a great laboratory environment there. And I think these elements that you're pointing out are so important. Being able to talk to each other like your colleagues, being able to spend time together, have fun, and maybe talk a little bit about things outside of science can be really refreshing, and it's a good way to get away from it for a little while.
Absolutely. It's a lot of fun.
Well, I like it. It has been fantastic to hear more about your lab and some of the work that you guys have going on. But I want to talk next about some of the big questions that you haven't yet had a chance to answer. So, Pat, can you tell us if things like funding and staff and technology and feasibility out the window, not a problem, what is the one question you would want to answer?
So as I was mentioning before, one of the things that we've noticed is that each person is really their own best control. And so that's a different way of doing science, that if you're studying heart disease, you might be able to get 10,000 people and get blood samples from them or give them a survey. But what we need to do that, but we need to do that monthly or weekly or annually on a longitudinal basis. Because what we'd like to be able to see is, can we see somebody develop diabetes? Can we see somebody develop colon cancer? And so what we really need is to develop some longitudinal cohorts where we track people over the course of their life. And we know that a lot of these diseases happen at a low frequency, thank goodness. But we need to see people develop these diseases through the lens of their microbiome. And so that's something that is no doubt going to be very expensive, but I think is just critically important to develop these time courses where we can watch people's microbiomes change over time and then associate those changes with changes in their health. And that's something that I think people are slowly thinking about. How can we do the fundraising or ask questions in a thoughtful way so it's not so difficult? But that's really a big challenge and a big need in our area of research.
So do you foresee cost to continue going down in the technology and the sort of resources needed to do this sort of thing?
Yeah, but we're greedy.
Right.
The big cost to us has always been DNA sequencing. Mm-hmm. I don't know the exact numbers, but it's so cheap to do DNA sequencing now compared to 15 years ago when I was in grad school.
Sure.
Our reaction to that is, well, let's sample more people.
More often, yeah.
More often, right? So it's not like we have this fixed design that we need to implement. We need to compensate people for their time and keep track of the people over long periods of time. But there's also a lot of computational expenses and doing the analysis. So we hope that things get easier with time, but I think some of those costs are just fixed and just going to be hard because we're working with human subjects.
Sure. Well, wonderful to hear about this question that you are ever working towards the answer to. And I think in addition to talking about these big questions, you get so many great opportunities when you're with your mentors, when you're with your peers and colleagues to get advice and to share advice. So I want you today, Pat, to share with us one of your best pieces of advice that you think you've ever received.
So this was a very subtle piece of advice where nobody said anything, but I just kind of watched things happen. When I was a postdoc at Wisconsin, I had a friend who was a assistant professor in the law school. He was kind of an odd duck there, and he was an empiricist in a law school. And so he would study bankruptcy law, and he'd do a lot of machine learning and big data type approaches that are pretty common today, looking at, say, bankruptcy or immigration law. And he got a grant and all these things that to me would just be like, oh, right, perfect. But he wasn't the perfect law school professor where you write your paper a year and you do whatever. And he got denied tenure. Oh, no. Which just like was crushing to him, I know, and me and other people around him, because it was just like injustice, right? So he went on the market because he needed a job. Sure. And he got hired at another law school with tenure. Wow. So, I mean, nobody said anything to me, but to me, that was just like, if you do everything you need to do, get grants, you write papers, you can control the things that are in your hands to control. If UMass, if Michigan, if they don't want you, somebody else will. Thankfully, I didn't have to test that hypothesis, but that gave me a lot of solace to think about. I just got to put my head down and do my work. And if I do good things, hopefully good things will happen. And my friend is very happy where he's at now, and things obviously turned out pretty well from there. And just that idea that to control the things we can do. If Michigan doesn't want me to work on my grants here, I'm sure somebody else will, and things will hopefully work out. So that's been the piece of advice that I've picked up on and really motivated me as I was a postdoc and then an assistant professor, worrying about tenure and kind of how to get through the ranks.
Well, this is really empowering and I think really helpful for some of the people out there who might be going through these career transitions. And I think things, like you said, will work out if you put in the work, if you do everything you're supposed to do, even if they don't work out exactly in the way that you expected. So great lesson to share. Now, Pat, is there any other last piece of advice or any last note of inspiration that you would like to end our interview on today?
I think the big thing in my career that's really made the difference was that engineering training and the ability to think quantitatively. And it's really all because I took one programming class as an undergrad at Cornell in a language called Pascal that nobody wants to use. That's just horrible. I don't expect people to go out and get an engineering degree, but I think the thing that people just need to do is to develop some type of programming skills and some confidence in working with data in that type of environment. We hear a lot of people talk today about big data. It's in the news. It's in the popular news. It's not just scientists that are talking about big data. But 10 years from now, we are not going to have less data.
Right. It's just going to get bigger.
We don't know what to do with the data we have now, right? Just imagine 10 years from now, it's just going to be worse. So I think those people that are able to develop the computational skills today will be so much better served in 10 years than if they don't. And I hear from a lot of biologists, I'm a biologist because I hate math. And that's like the worst thing you can say to me. I mean, it's the worst thing you can say for your career probably too. But Developing those computational skills is really critical. And thankfully, there's a lot of great organizations out there that are set up recognizing this. And so there's a group called Software Carpentry or another group called Data Carpentry. They go around and they do hundreds of workshops a year across the world. They're 2 days long and they help people develop some of these computational skills to at least get them going. So I kind of freaked out some grad students a couple years ago. I was invited back to Wisconsin to give a talk. And when we go give these talks, we typically have lunch with students. And the students asked me the same question you asked me, and I told them exactly what I just said. And several of them turned white and because they're just like freaked out. And somebody emailed me following up, what do I need to do? So I think that is like a really critical thing. There's a lot of people that can do genetics. There's a lot of people that can run gels and do Westerns and do whatever it is people do. But the ability to then tie that in with something computational or to merge these approaches is really powerful and would make a lot of difference to people in their careers.
Definitely. Well, Pat, thanks for sharing this piece of advice and some of those resources there. And I totally agree with you. These statistical and computational skills, you cannot undervalue them. I think they are so important for allowing you to continue to keep up with the advances in those areas and then also be able to work more efficiently in what you're doing if you can do some programming.
Absolutely.
So phenomenal advice for our listeners out there. And Pat, thank you so much for joining us today and sharing your story. Can you tell our listeners, though, how they can reach out if they want to learn more about you and your work?
Sure. So I'm fairly active on Twitter. My handle is at Pat Schloss, first and last name fused together. And you can also get me by email. My email address is pschloss@umich.edu.
Perfect. Well, listeners, definitely reach out if you have any questions or want to get in touch. And Pat, thanks again. It was a pleasure to have you on the show.
Oh, the pleasure's mine. This was a lot of fun. It was great.
Wonderful to hear. And I'm glad you had a good time. Listeners, I hope you enjoyed our conversation as well. We'll see you next time on another episode of People Behind the Science. Your voyage to the lives of today's exceptional scientists has just begun. You can find everything we talked about today, including our guests' favorite books, biographies, photos, and more, when you visit us at www.peoplebehindthescience.com. I look forward to seeing you next time on People Behind the Science.